Okay, hello everybody. Welcome back to the next installment of the OpenWorm Journal Club. Today we're talking about closing the loop with brain cells and behavior, and we're reviewing some recent work from Beer and Izquierdo. We have a great set of panelists joining us today as we review these papers. So let's go ahead and introduce them real quick, and we'll get onto our presentation. So let's start with Michael Curie.
Hi, I'm Michael Curry. For the past 10 years or so, I've been a programmer and entrepreneur in Calgary, Canada. And for the past 2 years, I've been volunteering my time with the OpenWorm project, working on movement validation, the sub-project to validate the movement of our simulation is correct. So I'm very much looking forward to today's presentation.
Great. And let's go to Shrikant Chalasani.
Hi, I'm Srikant Chalasani. I'm an assistant professor at the Salk Institute in La Jolla. I do have a nickname. Everyone calls me Shrek. And I study neural circuits and behavior in the worm using both genetics and computational.
Terrific. And Netta Cohen?
Hi. So I'm Netta Cohen. I'm a professor in a computing department, computer science department at the University of Leeds in England. And I'm interested in the neural control of behavior in the worm. We do a lot of computational modeling and some experiments and try to combine them, close the loop between experiment and theory.
Excellent. And we have the benefit of the authors of the papers that we'll be talking about here. Actually, we'll talk about some of Netta's work as well directly. So we have Eduardo Izquierdo and Randall Beer here joining us as well. So could you guys just briefly introduce yourselves as well?
My name is Eduardo, and I've just recently started my assistant professorship here at Indiana University, and I focus on, again, the neural basis of trying to understand the neural basis of behaviors in organisms using computational models, and part of the focus has been on understanding CA.
And I'm Randy Beer, a professor here at Indiana University, and I'm very interested, sort of broadly speaking, in what I call brain-body environment systems, in which C. elegans is a very nice, I think, experimentally approachable instance.
Terrific. And last but not least, we have our assistant here who's been making all this possible, Ben Shi.
Hi, my name's Ben. I'm a first-year PhD student at UC San Diego, and I'm working in soft robotics. I'm interested in the intersection between biology and robotics, so that's why I'm here to learn about the OpenWorm project.
Terrific. All right. Well, thank you, all of you, for spending this time here. So what I've done is I've gone ahead and compiled a presentation that's going to be looking at these papers here. So as I was mentioning, for the panelists that are here in the Hangout, please feel free to stop me, especially if I say anything that's incorrect, or if you'd just like to add or comment or ask a question. And as well, if you are viewing us here on YouTube, feel free to send in questions by tweeting to us or commenting on Facebook or Google Hangout or any of the various platforms that we have, and we'll try to get your question in here over the course of this next couple hours. Okay, so let's go ahead and get started. Okay, so here's the overview of what we're going to do. So first of all, in the sort of standard journal club style, I'm going to introduce some necessary concepts for getting ourselves understood and clear on understanding the results of Izquierdo and Beer 2015. Then we'll go ahead and review Izquierdo and Beer's 2015 results and conclusions, spark a scientific discussion, and then if we have time towards the end, maybe discuss some practical sync points with the OpenWorm project. So when we advertised this journal club, we actually pointed to two papers. So we pointed to this Izquierdo and Beer, integrated neuromechanical model of steering in C. elegans, and then we also pointed to this Izquierdo, Williams, and Beer, which described information flow through a model of the C. elegans klinotaxis circuit. And they are both great papers. As I was preparing materials for this, I realized that I could spend 2 hours going through both papers, or spend more like 30 to 45 minutes going through the necessary information for one paper, and then also have a good discussion. So I will have one slide towards the end on the second paper as a teaser for folks to continue to explore it, but unfortunately we won't really have too much time to go through this one. But that does mean that we have more time to really do a good job of establishing what the background was for this paper, because this is actually resting on some other important work that we want to be sure to go through. I'll also say that these slides that I'm presenting right now, I will post the link following this presentation on the Google Hangout page and on social media, and everywhere there's an underlined reference like this is actually a live link to the paper. So you'll be able to follow along even with some of the references if you like to get the original sources for this. So this will be a dynamic version for you available after the presentation. Okay, so the Izquierdo and Beer paper that we are going to focus on actually relies heavily on two other works that I need to introduce a bit before we can really fully appreciate what was done here. And so those are, first, Izquierdo and Beer 2013, which is titled Connecting a Connectome to Behavior, an ensemble of neuroanatomical models of C. elegans klinotaxis, and Boyle, Berry, and Cohen 2012, Gate Modulation in C. elegans, Integrated Neuromechanical Models. So this paper heavily cites these two, and so we need to understand a little bit about both of these. Folks in the OpenWorm project are much more familiar with the Boyle and Cohen model, because we have used it and had many conversations with Netta Cohen. Haven't had as many conversations with Eduardo and Randall. I just got to meet Eduardo this year at SFN, which I was very grateful for. So let's dive in a little bit here to understand this. So basically, to be clear, what this paper is a combination of this minimal klinotaxis neuronal network from the 2013 paper and the integrated neuromechanical body model. Scientifically speaking, they have taken this neural network and combined it with the body model and neural network that was in this paper. So let's just briefly answer the question for those of us who do not live in heavy animal behavior land all the time, or not in the C. elegans field. The word klinotaxis may be a little unfamiliar. So it's if you know your Greek and Latin roots, so taxis is a word that refers to the movement towards towards something, to orient towards something. And the klino part may be less familiar, but it involves a wavering side-to-side motion of the head occurring as an organism moves forward in response to a source of stimulation caused by the alternating reaction of sensory receptors on either side of the body. So it means, so the klino part specifically means the moving side to side. This is related to another word, chemotaxis, which just refers towards the movement towards or along a chemical gradient of some kind, but that doesn't capture the side-to-side motion part that's captured in the klinotaxis word. So that's actually a key part, because later when we get to some of the results of the model, the fact that the head is in motion actually plays an important role in understanding the model of the neural circuit. So that's klinotaxis. And so that word klinotaxis, has been around for a little while. Although it actually has an older history, I tracked down this old paper by Ward in 1973, where he originally showed, chemotaxis and then also klinotaxis, along the way, where he basically took a dish. So this is a petri dish of our friend C. elegans, and the one on the left has a chemical gradient, towards the center, sort of a salt gradient in the center. And if worms are placed around the edges at these 3 points, they will tend to move towards the center, up the salt gradient. C. elegans likes to be towards a higher concentration of salt, because bacteria that they like to eat will give off a bit of a salty chemical gradient. But if there wasn't a salt put in the middle and the worms are put on these 3 points, then they don't move towards the center, and they just kind of hang around in their local networks and just kind of run around. So so that's basically your chemotaxis and klinotaxis. Okay, so now we said that the paper is combining a minimal klinotaxis neuronal network with this body model. So what is this minimal klinotaxis neuronal network about? So this now goes back to the work of Izquierdo and Beer in 2013, and they started with a maximal network of the C. elegans, and they pruned it down to this minimal klinotaxis network. So again, guys, here, if you know, I'm going to do this rather rapidly, so if there's any key details that I'm missing out, please chime in. I don't want to mischaracterize any work here. But from my understanding of this, so we started with essentially the whole C. elegans connectome network. Those of you who are familiar with you know, modeling worms will know that we benefit from the foundation of a fairly complete connectome representation of the nervous system of the C. elegans worm. And so this is a sort of representation where they've aligned the chemosensory neurons on the axis up here and the motor neurons on the bottom here in black. And then in between, they've outlined the rest of the network with black lines being chemical synapses and red lines being gap junctions. And so they started with this from the beginning, and they pruned the network using path length as a criterion and maximally connected you know, network to itself as a criterion. And in this 2013 paper, they pruned this down to a much more manageable network for analysis, which consists of these neurons here. And so you'll see this. So this will be the same network that's used in the paper that we talk about. So it's pretty important to understand. So we start with the ASE neurons, the chemosensory neurons here at the top. We go through 2 levels of interneuron processing through AIY and AIZ. And then we come out at the bottom to the 4 motor neurons here that are with the SMB family, the ventral left, the dorsal right, and the ventral right. And it keeps, this diagram keeps the chemical synapses and gap junctions convention. And so this is then a minimal klinotaxis network. So another thing to say is, so why is this the minimal klinotaxis network? So it's important to note that these are not just any, you know, these are not just any sensory neurons, but these are neurons that are sensitive specifically to chemical gradients. So ASE being you know, strongly implicated in that. If you knock out ASE with the laser, you get you know, inability or drastically reduced ability to move towards chemical gradients. And then the neck motor neurons here, obviously there's a lot of motor neurons you know, in the worm beyond just these 4. You know, many of them represented in the integrated body model of Boyle and Cohen, but these are chosen as these are primarily implicated in steering, if I understand correctly. If these are ones chosen because of that. And guys, do you guys want to say anything more about SMB as the motor neurons here, Eduardo and Randall?
So I think the main reason for choosing SMB at that point was because they were implicated in ablations to SMB resulted in loopy behavior. So sinusoidal movement that was more constrained. The amplitude of the movement changed in a gray paper in PNAS. So it's not directly implicated in steering, but it was a hypothesis for if there's a neuron, if there's a neck motor neuron, that can, when killed, change the amplitude of modulation of the wavelength of movement, and that the modulation of that neuron, of the activity of that neuron, could be linked to steering.
Okay. Okay. Good. So that's a useful clarification. Yeah. So to point out, you know, what this is not, or not attempting or purporting to be, is, you know, a complete description of all the motor neurons, which are also just involved in just general forward locomotion. So the idea here was, because they're searching for this klinotaxis network, they want the subset of neurons that are specifically going to translate information about a changing chemical gradient into a different direction of the worm's movement. That's what this is intending to capture, if I understand correctly.
Yes, and I mean, part of the assumption that is going in is that we're trying to see if changes in just the neck motor neurons, which are very different than the rest of the ventral cortex.
That's right.
If changes in the head, so SMB is a neck motor neuron.
That's right. That's right. And you guys point out in your paper that with the worm, it's essentially the body is following the head, or the idea is that the head changes its angle somewhat to orient towards the the gradient, and then the rest of the body sort of follows along, so that the rest of the motor neurons may just be the kind of the engine, the gas, and then this is kind of the steering wheel, to use a very coarse description. So, okay, excellent. So, as we like to do in the OpenWorm project, I did throw the minimal klinotaxis network into our OpenWorm browser, and I just visualized the left-hand side of the network. But this is our little 3D browser, and I won't go through the playing with it dynamically thing that I sometimes do. But basically, so you got ASE is up here to kind of visualize this. So this is the head of the worm. We're looking at sort of right and left mirrors of each other. So this is the head of the worm up here, and so ASE, the sensory neuron, basically goes all the way out towards the snout of the worm here up in the head, and so it's well positioned to receive chemotactic signals or concentration gradients, and then it goes in through this network into AIY and AIZ, which is near the nerve ring. And then SMB will travel sort of down the body of the worm here, innervating, I assume, with muscles as we go down the body of the worm as well. So this is kind of what it looks like in 3 dimensions, and so quite nice. Set of neurons. Apologies that the labels sort of cover up the cell bodies, unfortunately, down here where the action is. But yeah, so that's kind of what it looks like. Okay, so some key points that are pointed out in terms of the dynamics. So I'll spare audience right now from the equations, just because we're trying to get through this. But I'm going to characterize what the dynamics of the neurons are in terms of the neuronal model that was used for this, also because it's important for the next step. So sensory neurons here are described as on or off. That kind of integrate over a history of the chemical gradient concentration and include a rise and decay constant. So I believe the left and the right are opposites of each other. So one is going to be active when it's on, and the other is going to be active when it's off, or when it's sort of receiving a chemical signal, it's more excited, and the other, when it's receiving a more chemical signal, it's less excited. And so these are used as the sensory neuron models. The interneurons are passive nodes, whose membrane potential is basically representing the summing up of the inputs. So those are the AIY and AIZs. The chemical synapses are releasing neurotransmitter tonically or constantly, and their output is a sigmoidal function of the membrane potential, and it also includes a bias term, which is related to, I think, the sensitivity of the chemical synapse. And then lastly, the neck motor neurons are the same as the interneurons, but they also have sort of self-connections, and they also include an oscillatory input that is, the assumption here is that there's an oscillatory pattern that is created by other parts of the network, which basically these motor neurons are sort of on the receiving end of, and so that's factored in here. So guys, is this a reasonable? I know, I mean, obviously this might be painful because the ultimate description of this really is the math, but at a high level, is there anything that I'm fundamentally leaving out about the dynamics?
Can you go ahead and say,
I just wanted to be clear. You say sensory neurons are on or off. That doesn't mean that their outputs are binary.
Yes.
Yeah, okay, just to be clear. It has to do more with their response to a change in chemical concentration, whether they're sensitive to an up step or a down step.
So maybe the wording is they're rectified, but they're graded.
Yes.
Very important. Yes, that's yes. Yes, the rise and decay constant, obviously, making it contributing to that grading. Yes, so that's important. They're not. Yeah, they're not. Okay. Anything else then?
The other point that you can make one additional point, which is that a sensory neuron, while giving you an increase in calcium signal as measured by to the addition or removal of stimulus.
Oh, hey, Shrek, is that you? Yeah.
Yeah.
Could you just speak a little bit closer into your microphone, because I think you're really
Okay.
faint.
Can you hear me now?
Yeah, that's a little better. Yeah. Mm-hmm.
Okay. So the point I want to make is that sensory neurons might signal both they're typically the data that's presented or published right now suggests that sensory neurons respond to either the addition or to the removal of stimulus. And the response is measured by increases in calcium concentration, measured by genetically encoded calcium indicators. However, there might be responses that are also showing decreases in calcium concentration. So you might have multiple signals coming out of the sensory cell that the interneurons are responding to, are integrated.
Okay, got it. Let's keep working on increasing your volume so it was a little bit better. But I think what I was hearing from just if I got it was you're sort of saying that that ASE is responding to the extra addition of chemical signal or the removal of it. So it's essentially responding to changes, and that it's mediated through the calcium. So what that does is it changes the calcium inside of the ASE cell, and that there may be responses as well downstream to the increase and the decrease of the calcium gradient inside the.
Right.
Is that
so?
Yeah. Oh, that's much better, by the way, with your. Yes.
Okay, so the point I was trying to make was people, when they report sensory neuron activity, people are reporting increases in calcium concentrations by sensory neurons.
Yes.
Very few studies are also looking at decreases in calcium concentrations because our indicators are not capable of picking up the decreases effectively.
Yeah.
And so the current experiments or all of the experimental data is mostly focused on the increases, but sensory neurons might hyperpolarize, and hyperpolarizing sensory neurons might also have information to the rest of the circuitry, particularly in a graded network where neurons are detecting both increases and decreases.
Perfect. Yeah, got that. No, I think that's really important. And yeah, so this actually, I just wanted to make a more general point. So this is going to the general conversation between experimentalists and theorists, and how obviously there as you're building a model, you try to take the best that you can from what we understand from the experimental results, and then you make some assumptions on top of that. And I think the great thing about bringing both sides in here is that we can discuss what are the common limitations in the data that sometimes that we need in order to improve our models. So yeah, that's very helpful, and we're gonna keep adding those in as we go. Okay, great. So these were the basic model here. And I should also say too, one of the important things about a model that has relatively few parameters is that it's computationally quick to run through and calculate, and this is going to be a big benefit for them in their next step, because so they have a strategy here after defining the minimal network. So the logic of this paper, as we've shown so far, is define this minimal neuronal circuit by mining the connectome. Then the next step, they use an evolutionary algorithm to find the unknown electrophysiological values. And then after that, they're going to characterize the resulting models that come out to see sort of which is best, or what properties the ones that are best have. So let me talk a little bit about this evolutionary algorithm. So the fact that this is a network that has sort of few parameters and is easy to compute is a key facilitator for actually practically being able to do this evolutionary algorithm. So let's talk a little bit about that. So actually this idea of evolving a neural network has been around for a little while. I dug in a little bit the references, and I found Randall, this paper by you and Dr. Gallagher back from 1992. And this sort of, this whole idea kind of warms my heart. It's something that's more new, I think, in the computational neuroscience field to do this. But I also, have had a past in artificial intelligence, and so this idea, of combining an evolutionary algorithm and using it to modify a neural network is something that has a really cool history that is sort of also derived from the artificial intelligence field. So the basic idea captured in that paper, and you know, there's a whole literature on this, but just as one exemplar, is you know, so in this paper they were working on a simulated insect body model like this that had different you know, motor controllers for legs that would move around, and so they had a neuronal network that would basically control the movement of the leg, and so this was a simple neural network that would control sort of the movement of the foot, and one that would be responsible for a backward swing and a forward swing. There was an angle sensor that was involved in here. These are, of course, you know, fairly idealized neurons, neuron models at this point. But anyway, and then they could combine them together in a network across, you know, articulated body of an animal. And what they would do is have different of these parameters that were put under the control of a genetic algorithm, which could search through the space of how these were wired up and what kind of dynamics they would put together. And I think there's this really good illustration of work like this that generated this, you know, rather famous movie in you know, 1994 by Carl Sims, along this sort of same idea where they evolved virtual creatures. So this combined a three-dimensional view. So I just kind of wanted to show this real quick here. So apologies for the low resolution. I don't know that higher resolution versions are available. But what you should be seeing here are sort of clips of what look like just three-dimensional, like three-dimensional shapes, blocks basically, you know, cubes and rectangles, which are involved in various kinds of motion, either sort of swimming motion or walking, or what appears like walking motion. And even though they're very simple shapes, they have a variety of behavior. And what this is showing is not the result of a human being designing these different shapes, but in fact both the shapes and the sort of underlying control system for these were evolved in a computer using a essentially genetic algorithm of some kind. With right. So the way that it worked is that you start at you know, at time 0 with a shape that is connected and doesn't really move very well. And then a key point here, which is also, you know, going to be reused in this paper, is that in the first generation, you know, it doesn't move. Then you change some parameters and you go to the next step, and you see if it does a better job of moving in a direction that you're interested in. And maybe some of them move a little bit, and then you pick the ones that do the best moving in that direction. And then you evolve your next step. You change your parameters again. You get another generation, and then you see if they move a little bit further down the path. This is actually showing something that's a lot more like chemotaxis, with movement towards this little red dot here, which actually moves around. So so this obviously is not you know, not showing you know, a worm, and is also leaving out you know, obviously many aspects of the details of the cellular biology. But what this is intended to show, and I'll pause it before we get to that one, what this is intended to show is what is possible just purely from a computational perspective at training up a little agent that's able to kind of move through a space you know, over time. So this is an important fundamental idea to understanding what happened in this Izquierdo and Beer paper. So for everybody else that's in the room, is anybody have any questions about this so far? Okay. Cool. So let's keep going. All right.
I just wanted to make one comment, if I could.
Oh yeah. Yep. Comment. Yep. Great.
What's important for the C. elegans work is not the evolutionary algorithm per se, but the fact that we're using some kind of stochastic optimization technique, and there are different ones available. The key thing is that we're trying to search the space of what's unknown, constrained by what's known, and then study the structure of the resulting partially constrained models that we get. And it's just that evolutionary algorithms is a particular technique that we're most familiar with.
Perfect. Yes. Yes. Good. So good clarification. So specifically, so I dug in through the paper, and so they picked particular parameters that were in this minimal klinotaxis neural network from the dynamics, and open those up to evolution. So it's not everything. Yeah. So that's important to point out. So it's a limited set of parameters, and they point out in the paper that these parameters have not yet had the benefit of being able to be measured experimentally. So they really are doing this to fill in gaps here. And Netta, did you have a question? I think maybe you were trying to speak, but you were muted, or a comment?
She's still muted.
Netta, you're still muted.
Yeah, we can see you, but I think you just got to hit the mute button. So it's should be if you roll over the Hangout.
Okay.
There you go. Yep, there you are.
Okay, so I was just going to make a comment about the distinction between the two aspects of the sort of agent evolution, which are the situatedness and the embodiment. There's two completely different aspects here. One is how does the body influence the control and the behavior, and one is how does the interaction, the closed loop interaction with some complex environment. And they can be treated individually or together.
That's right. That's right. That's important. Yeah. So this whole other idea of embodiment and why you might use embodied systems. There's actually a whole lot more to discuss as well. And but I think one of the to say critically here, nervous systems control bodies, and this whole discussion here is we're trying to figure out how to go all the way from the activity of cells to behaviors. So having a body that's appropriately that you both exists and is situated in an environment, right, are two key aspects of fully understanding the relationship between the cellular activity and the body. Yep. Perfect. Okay, so there's strength of, so just back to the parameters here. So strength of connection from the neurons to the muscles, the rise time, decay time of the sensory neurons, the strength of the connection to the oscillatory component that I mentioned in the neck motor neurons, the strength of the connection from the, and this is just replace this with the sensory cells to the downstream neurons, and the strength of the chemical synapses. And then there's this bias term that's also set to evolution that shifts the sensitivity of the synapse output, sort of the sensitivity of the postsynaptic neuron to the presynaptic neuron. Okay, and then important to note that the fitness that's used is this idea of a chemotactic index, which is a function of essentially the average time that it takes for the agent to move to the peak of chemical gradient, where faster is better. So what they're doing is they're basically evolving a network to have the dynamics that are going to maximize the time that it takes to move the body towards to the peak of the chemical gradient. Did I get that right? Is there anything else to add to this, guys?
Yeah, it minimizes the time, which maximizes the chemotactic index.
That's right. That's right.
And one thing that I wanted to add was, so the main point is, so we do have the neuroanatomy that we can get from the connectome. I think the main point is that electrophysiology hasn't been done to know the strength of the connections between some of those interneurons and neck motor neurons. So the idea is, or the intrinsic dynamics of each of those interneurons, like AIY and AIZ. So the point of the evolutionary algorithm is to search for different possibilities, different configurations of strength of synapses and gap junctions that will lead to the correct type of steering, which will reflect in the chemotaxis index.
That's right, and this idea of generating multiple models is also something that you guys cite work by Eve Marder that have done similar work in stomatogastric ganglion of the crab and lobster, and so this is a practice that also has a history in the literature, specifically in neuroscience as well. And so you're kind of so it's kind of a nice combination, I think, of this sort of AI perspective, and then also something that's been used specifically for neuronal circuits to tune parameters. Okay, so then, so before I go to the results then, so just as a quick summary, right? So they start with a maximal network. They prune it down to this structure that includes cells that are necessary for chemotaxis. The dynamics of those different cells are this model here, which is chosen based on data that's known about the way that these neurons should operate, and then it's subjected to this evolutionary search along these parameters and using this fitness function. Okay, so that's.
Let me make a correction, Steven.
Yes.
So the way we arrived at the minimal circuit, it wasn't because those neurons were necessary. So some of them are, the chemosensory neurons are, but the reason for the minimal circuit is that they provided the strongest pathways, the strongest and shortest path from chemosensory neurons that were known to be relevant to neck motor neurons that we hypothesize could be important for steering. So again, it's just shortest and strongest paths. And what we show with the maximal network is that there are many paths because the connectome is very densely connected.
That's true, and you also, that's right, and you also made a point, I think, in that paper to show that there was this intermediate network between the minimal and the maximal.
A lot of intermediate networks, depending on how tightly you draw your assumptions.
Right.
So it's a starting point, not an ending point.
Right, and then in your 2015 paper, actually, so I just wanted to drill in on one thing. I noticed that there was this dissertation by McCormick, which you guys pointed to in your 2015 paper, which you said had done some additional experimental validation of this minimal klinotaxis network. And you said a few things about, like, it also showed an asymmetry. Could you guys say a little bit more about that validation piece? I was curious about that one. I didn't have time to dig into that dissertation completely.
So they have been doing experiments. So Kat was a PhD student in Sean Lockery's lab, and she had been doing experiments using on klinotaxis with the worm. And one of the things she found was that they did perform ablations on all of these interneurons, AIY and AIC and SMB, I think.
Individually, rather than by class, yeah.
Yeah. And they found, right, individually, so left and right. And they found all of those neurons, and other neurons too, obviously. But then they found those neurons, the ones that we have found in that circuit, to be necessary for the behavior. Mm-hmm. In the specific context that they were testing it. They have this klino chip that they pin the worm down and leave only the anterior most part of the worm, and they try to see how much it moves from one side to the other.
Cool. Okay.
But the asymmetry had to do with the consequences of ablating a left or a right member of a class. They were different, and that's the point we were making, because we found the same distinctions, the same left-right asymmetries in our analysis of that model.
Right. Could I ask a question about that?
Absolutely.
In your model, you have gap junctional connections between the left-right AIYs and between the left-right AIZs.
Yes.
And so a lot of people will generally hypothesize that those are synchronized, and yet in order to achieve the symmetry breaking, you would need that to be violated, as you do in your circuit. Or how does that exactly work?
Well, so the strength of the electrical connections is the same in both directions. It's a bidirectional connection.
Right.
But the chemical synapses are not symmetric. So you can have an ASEL to AIYL strength that's different from an ASER to an AIYR. So there's asymmetries there. The other thing is the electrical connections asymptotically will tend to synchronize. But that doesn't mean that transiently the effects are going to be synchrony. So it can be timing dependent as well. And also, electrical connections operate on the membrane potential, whereas the connections the chemical connections between cells have this sigmoidal nonlinearity effect. Right?
Right.
So all that plays into these asymmetries. Sorry, go ahead.
So I'm trying to understand what you're saying. So, I mean, there's also work in the past where sufficiently weak gap junctions have been used to get anti-phase synchronization between neurons. Famous work by Sherman and Rinzel in 1982.
Yes.
So the question is, in your case, are they indeed in anti-phase?
No.
So you're asking if they're effective? No, I mean, no. I guess the short answer is no.
And the only thing that has a phase, I guess,
Is the SMBs.
are the SMBs. So in our case, it's SMBs that are innervating the muscles, and in some ways have this reciprocal input from the muscles if the head were moving. So they both receive. The assumption is that they receive an input and then they generate an output to the muscles. So they're the only ones that have a phase. The AIY and AIZ are just receiving sensory input.
So the way I would interpret that, and please correct me if this is wrong, is that you have a CPG that sets the phase, driving the SMBs, and then in addition to that, the sensory motor pathway is now modulating that to affect steering.
Yes.
Yes, except I wouldn't see it as, I don't think it's restricted to a CPG.
That's true. Yeah.
In the specific
Yeah.
Just to clarify my comment about the electrical connections, what I meant was if you have two neurons electrically coupled and you step one to some held membrane potential, the other one is going to equilibrate with it asymptotically. The out-of-phase thing you're talking about is when you couple two oscillating cells, which of course can have more complicated phase relationships.
Right, but the reason I'm suggesting that is because the sensory input could be oscillating since the worm is wiggling.
It is, yes. Yeah.
And so not to overcomplicate this actually, but this is the one slide from the from the second paper that I had pulled out, and it does relate to this. And this may or may not, it may not directly relate because this is a diagram of mutual information, but just because this seems like a good point in the conversation to mention it. So this second paper about information flow, now that we've explained a little bit about this circuit, this is the same circuit again, just with slightly different schematic layout, but it's the same one. They're basically looking at the, what can be said mutual information-wise from one neuron to the other, where mutual information is the reduction of uncertainty about one random variable given the knowledge of the other. And so this I thought was just a useful, this is their first figure, or this is the second figure I think in the paper, but this is the first one, where they're looking at, you can kind of see the course of events, if you will, from the change in a chemical gradient in this first line, the activity of, the mutual information of ASE, and then how that sort of flows into AIY, AIZ, PG is this internal sinusoidal input, and then how that has an impact on SMB. So I'd assume that, I guess to Netta, to your question, so the red and the blue on the AIY and AIZ lines are the left and the right. So if the gap junction was very strong, well, yeah, if it was like a direct connection, if it was like the same thing, then I guess if I'm interpreting this diagram right, guys, then these lines should essentially always overlap, and that there would be no, or they'd be flat, I guess, right? They'd be flat because there would be no mutual information different between the two. There would always be the same. But the fact that there is an asymmetry in here is kind of showing that is probably a weaker gap junction connection of some kind, at least transiently. Is that?
I don't know how much you want to get into this. It wouldn't be flat because what this is showing is mutual information about the concentration step. So I think what you're saying is,
yeah, right. Okay, yeah.
If the two cells equilibrated in exactly the same way very quickly, then they both carry the same information. And the fact that they don't is related to this asymmetry we're talking about. Yes. Another way of looking at it is later in the paper, we look at the transfer entropy that goes across that, essentially the information flow that goes across the electrical connections, and it's asymmetric. So there's more information flowing in one direction than in the other direction. And that's kind of interesting because your naive assumption would be as I said earlier, that the electrical connections are just always essentially doing the same thing in both directions. And they are, in terms of the current flow, the underlying Kirchhoff laws, but the consequences of that in the context of a network are not always so simple.
Right. So that's right. Yeah. So it's mutual information about the internal the stimulus, and so right. So if AIY and L and AIYR were doing the same thing, then this should be the same. It would be the same curve, basically. Right. Okay. So right, and that is one of your, I think, predictions that you make in this paper specifically is that the integration from ASE to AIY is only happening in one. That is a symmetry that you'd find in the animal. If you recorded from AIY, you'd see the integration happening in one but not the other. Does that characterize it correctly?
I'd say that's a little strong.
Yeah. Okay.
What we can say is that given the assumptions that we put into this model,
right.
including the particular neural model and parameterization, the restriction to the minimal circuit, blah, 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 the whole set of assumptions, we consistently find that pattern.
Okay.
Whether or not that's going to be found in the animal is a little strong, I think, at this point, because the model is so simplified. What is interesting, though, is what Kat McCormick found was that there are asymmetries in effect of lesioning AIYL versus lesioning AIYR, and so it appears in the animal that they're also not operating as just two copies of the same thing, but they have their distinct identities in the circuit's behavior.
Got it.
A little more qualification, but yeah.
Okay, yeah, no, that's important. And so actually, so this question for Shrek as well, since I know that we're getting close to when you have to take off here. So when have you seen these kinds of analyses on your side just experimentally in terms of looking at just thinking about how downstream effects of change in ASE affects various interneurons down the road? Is this a kind of analysis that's open to additional experimental verification or just gathering more data along these lines?
Yeah. So this is a really interesting model. I think the experimental data so far, I would say, is inconclusive.
Mm-hmm.
So we've done quite a bit of imaging at the sensory neuron level, and we know that both ASEs and a bunch of other amphib cells will respond to salt stimuli, depending on concentration of the salt and depending on prior history and other context of the animal. At the interneuron, we see that both the left and the right neurons respond to the addition of salt. So, for example, ASEL is responding to the increase of sodium ions in the stimulus condition, in the stimulus. Now, you'll see both AIYL and AIYR responding to ASEL activity, and similarly, AIYL and AIYR respond to ASER activity. So, however, these responses that one is measuring in AIY are occurring in the process where you're measuring changes in calcium at the level of a bulb in the neurite, which has a lot of synapses, and I think it's a little bit hard to separate out presynaptic and postsynaptic information in AIY.
Mm.
The other interesting part about it is that, at least in regular chemotaxis assays, AIZ seems to be the critical neuron for chemotaxis behavior. AIY you can potentially dispense with, so even if you get rid of AIY completely, that is both the AIYs, you're still able to get there. It might take a little bit longer, but you still find the point source of the attractant. AIZ, on the other hand, you get rid of AIZ, the behavior falls apart. So it seems like AIZ at the second stage is integrating additional information, perhaps from AIB and AIA, or maybe other interneurons, or maybe other sensory inputs to get you to AIZ. Now, interestingly, AIZ itself does not respond to step changes in stimuli. So the general assumption there is that perhaps what AIZ is doing is responding to gradients of changes in stimuli, whereas AIY and ASE and all these other cells maybe are responding to step changes as well. So chemotaxis might be a little more complicated than we can figure out experimentally yet, which is why we need more models to help us guide what experiments we should be doing. So in fact, what would be interesting right now, I would feel, is if we could measure AI-Y responses, I mean either the left and the right interneuron responses in the presence or absence of AI-Z to the two different sensory inputs. So that's an experiment that we can do.
Cool.
So we would learn something about what these two primary interneurons are doing, as well as what's going on downstream.
And just to comment, the whole point of the methodology we're pursuing is that we would expect to revise the constraints in the form of the model as a result of such experiments, and then we would be able to re-optimize it in the presence of new constraints.
Exactly. So I feel like this interaction of, experimentalists have kind of stayed away from modeling because we're a little bit nervous about it. But I think it's important for us to talk to you guys so that you can tell us what solution space we should be spending our time in. We could be off in like Never Land with all of these things.
As can modelers, definitely.
So I'm really excited about this one. So I read the paper. I think it's really cool. I like the idea that you have two layers of interneurons. That there is a primary layer that is perhaps getting information. Although the experimental results would say that apart from ASE, perhaps AWC is also playing a role there. And AWC is telling you more about gradients in salt concentration. And AWC also synapses onto AI-Y, so you have additional inputs coming into the AI-Y interneuron into layer 1. And in layer 2, I think we're off to the races. There are a lot of things out there that we just don't know.
Yeah. So some of our less than minimal models that we talked about in the 2013 paper definitely show other pathways to these neurons. And in fact, Kat McCormick's dissertation also looked at least one other neuron that the ablation study suggested was important.
I think it was
that's not included in our minimal model.
Yeah. So the one challenge that I think is becoming more common across the experimental model experiments right now is that the cell ablation experiments had defined necessity for the behavior, but in wild-type animals, additional neurons seem to be playing a role. So there are many more neurons that are participating in processing the information and integrating the information than had been previously predicted from all these cell ablation studies. And so I don't know if there is a quick and easy way of looking at the entire circuit. So we've been trying those experiments right now, and we find it completely fascinating. So I just want to bring it up to the group to think about. So what we have done is localize the calcium indicator to the nucleus of all of the neurons in the animal. So of the 160 neurons in the head of the worm, we can see about 110 of them in our imaging setup. And if you compare activity from different animals, you get different sets of activity patterns. So every single animal to the same stimulus gives you a different set of activity patterns. Maybe we haven't figured out what is common across them, and because we're not doing it, we're not segmenting our data in the appropriate fashion. But these 110 neurons give us whole different patterns out of them, even with things like salt stimuli or odor or food or any of them.
Which is another reason why we like this ensemble modeling approach. Rather than thinking about a single unique model, we can't forget, and this was Eve Marder's point, that there's considerable individual variability from animal to animal. And you're not going to modeling an average animal may be completely useless. You have to model the individual correlations that exist in particular animals. So in a sense, our ensemble is looking at that kind of stuff too. Although we have a long ways to go until we have a realistic enough model to say something tight experimentally. We view this as a methodology and an ongoing loop than anything else. We're not sold on this particular model really, except as a opening salvo.
Sure, we love it. I love it.
It's a strong one. Terrific. Good. Shrek, I want to make sure that you had a chance to say, any thoughts on your mind here before you had to take off. I know that you're about to.
So
any last parting words?
Right. So for me, I think it's a really exciting start. I want to continue these interactions. I mean, I usually talk to Tatiana Sharpie up in computational neurobiology here, and we've been looking at mutual information in the local search circuit as opposed to the solve circuit, because in that behavior, the animal is aggregating information over many minutes about variability in the food environment, and then using that information to drive behaviors that also last many more minutes, many tens of minutes. I think the models are awesome. So I would, more kudos to you guys, because we need to know what kind of solution space we should be probing in our pan-neuronal or broader experiments. Experimentalists are shifting from looking at one neuron at a time and one circuit at a time to now studying multiple circuits and multiple neurons. And so as we step into this world gingerly, we're we're hoping that the theoreticians will guide us in getting to the right kind of experiments to look at. Of particular interest would be identifying nodes that are relevant, or nodes that would be super useful for processing information. So, for example, AIZ might be one of those nodes. SMD might be another node that is absolutely critical for many types of behaviors. RMD is another neuron that also has this bilaterally asymmetric connections with muscles. Those might also be key motor neurons there. So my feeling is that these analyses kind of get us on the right path to look at what neurons we should be focusing on, because even in a simple system like C. elegans, it's still a difficult problem to solve experimentally.
Yeah.
Definitely. Shrek, I want to thank you for taking the time out to join us for this. So really appreciate it.
And I'm happy to answer more questions. I'm happy to talk to everybody. I also meet with Netta quite often, so I keep in touch with her. Thank you.
Excellent.
Thanks, Shrek.
Bye.
Okay, so just to pop up a level. So we've jumped around a little bit here in the presentation, but I wanted to get that in. So I think that was excellent. So okay, so we described the neural network. We looked a little bit at some of these results in that 2015 paper on the information flow, and that's a bit of a teaser. But I wanted to go back and just point out that, just get back to the other 2015 paper and get to the integrated neuromechanical model, which is the other big piece of that, and then look at those results. And then we can open it up to a more broad conversation. So this was just a figure from the 2013 paper that pointed out that if you do those things of selecting the minimal network and evolving the network, evolving the parameters, that you get behavior out that looks like this, where if we put our simulated worm here and we have it start to move along a gradient, it does that. The color coding here meaning the speed in which it does it. And so these are examples of the some of the best models that came out in terms of their ability to start. And they're started here at the different lines or from different starting orientations, just sort of showing that even if it's not pointed at it doesn't have to be pointed at the gradient in order to find its way over there, so that it does orient appropriately. And this is a zoom in for one of those. So excellent. Okay. So the second piece of this is what is an integrated neuromechanical body model, because the 2015 paper builds on that neuronal model and builds onto it this articulated model of the C. elegans worm. And this comes from the work of Netta Cohen, along with Jordan Boyle and Barry. I don't know Barry's first name, but in 2012. And this work sort of contrasts from the body model that was used in the 2013 paper, in that it has this full articulated body that in 2 dimensions, can show different postures of the worm in different scenarios. So either sort of example postures that come out when swimming in viscosity something like water, some kind of an intermediate gelatin, or sort of crawling through something with a viscosity approximately of agar gel. And so the model includes these black stiff rods that maintain a constant width between the two ends of the worm, and then the blue lines capture dynamics both of active muscles and passive forces as well. And there is a circuitry in here that involves the DD, DB, VD, and VB neurons synapsing onto essentially the body wall muscles. So this was the starting point. What had been described in the body model from the 2013 paper had been a point worm in 2 dimensions. So essentially the worm was always at a single point. And then in order to reproduce klinotaxis, the point sort of turns back and forth, representing the undulation of the worm at a given angle. And then that angle was being determined by the ratio of activations of the neck motor neurons, so allowing that angle to be larger or smaller depending on sort of the activity through the circuit, so involving the steering. So so it's so between these two, you can see this one involved posture, and it also involves the dynamics of the D class and the V class of motor neurons, which was not present in this one. But this one, focused on in 2013, it focused on essentially the steering. And so so now we have, finally, after an hour, we have the background, sufficient background to fully dive into the results of this paper in 2015, combining the minimal klinotaxis neural network with this integrated neuromechanical body model. And Neto, was that a sufficient description of, I mean, it probably wasn't sufficient, but was it at least reasonable?
I think it was short and to the point. I could mention the input that drives this is an on-off signal from a command interneuron, a forward locomotion command interneuron, which isn't in the circuit in the schematic, which is AVB, and that what propagates the neuromechanical signal down the body is a combination of the neural circuit and proprioceptive feedback in this particular model.
Perfect. And then there's also a contribution from stretch receptors in here, which I didn't mention, right?
That's what I meant. That's the proprioceptive feedback. Yeah.
Yeah. Right. Yes. Exactly. Perfect. Okay. Great. So then now the first figure of the Izquierdo and Beer 2015 paper, you will recognize in the A part. Sort of what they've done is they've reproduced the 2012 Cohen model. And what they've done is they basically bolted the steering wheel onto the engine. And they've done that by connecting their steering model that you see on the right now, which should be pretty familiar, the ASE to AIY to AIZ. It's just in different colors, but it's the same minimal model. And they've essentially, as far as I understand, guys, you plugged it into the connections that are up here in the head where the steering essentially happens. And you've experimented with doing it a few different ways to exactly which you've actually done that under the you've thrown that in under the evolutionary algorithm to determine how many and which ones to stimulate. But it's essentially plugging that's what you're plugging in. That's how you're making the connection between these two models. Is that about right?
Yeah, so let me see, to clarify, so in the Boyle and Cohen model, there's the dorsal and ventral muscle neurons are being driven by DB and DD.
That's right.
The ventral sides of it. And that goes throughout the body, throughout the length of the body in the BBC model.
Yes.
And what we're doing is we're leaving that unchanged. And in addition, so the neck muscles are receiving, in addition to the input from DB and DD, they're also being driven by the output from SMB. And so, and what we're playing with, so the green, those last neck motor neurons.
That's right.
So that's the only difference, is that now those neck muscles the ones in the boxes on the top left, are receiving input from SMB as well.
That's right. So at the bottom of the minimal klinotaxis circuit, this is representing motor neurons, and the output of these motor neurons is going to the muscles here that are also, in the Boyle model, are receiving the D class and the V class inputs.
Yeah, and the difference from the 2013 is the thing that's missing from that diagram on the right is that there's no central pattern generator driving oscillator, or oscillator in general, driving SMB. Now, so again, that's the whole point. So previously we were assuming the worm was moving in sinusoidal-like movement by driving it with a sinusoidal-like input. In this case, the thrust forward is being generated by the variant coil model.
Got it. That's a key point.
And the other point that, if I could clarify, it seems from this schematic, and I think that's correct, but correct me if I'm wrong, that the neuromuscular junctions from SMB, whether to the anterior 4, 8, or 12 muscles, are all synchronized.
Synchronized with what?
Anterior to posterior.
They're all going, she's saying they're all going at the same time. There's no delay or,
right.
Don't you mean there's
No.
Yeah.
That's correct.
Yes.
Not to say there wouldn't be, but in this model, it's just an additive interaction.
That's right.
Great. Okay, so then that's the combination between these two. So hopefully everybody gets that. All right. So then, in terms of looking at some results, so first some that are just sort of showing that in fact it does orient appropriately across chemotactic gradients. So these are just starting off by placing the worm in a position, and the color gradient back here is demonstrating the chemotactic gradient, and by placing it at this point, it finds its way again towards a peak, or it sort of orients itself in a direction of greater chemotactic gradient. Here is showing, depending on the kinds of gradients, whether it's a conical gradient or a Gaussian gradient, the different paths that the worms end up taking in order to find their way to the gradient peak. And this is just kind of quantifying the steepness, the quickness to which it sort of finds its way to high values of the chemotactic index.
Could I interject?
Yes.
About the methodology again. So it seems that we've skipped a step here, which is that in order to actually get to this point of running the simulations, some of the parameters had to be re-optimized.
Yes, completely right.
Yes.
So in some ways, so the picture at the top with so the picture at the top with the blue gradients. So because the body model now takes a lot more time to compute, we have the klinotaxis minimal model, which is again the picture on the right in the previous graph. But we're not using the same connections that we evolved previously on the point model. In this case, we are making those connections again starting from random, and we're putting it onto the full model now, and we're trying to see, using those three conditions that are at the top, which of those three,
that's the fitness measure.
That's the fitness measure. So
there's three cases.
So there's three cases where we're setting the worm, because we can't evolve it for too long. We have to make it very specific for shorter time periods. If it moves, and we put it in these three conditions just to see if it moves in the direction of the gradient, then we give it high enough points. And it's only once those parameters have been found again, those setup configurations of the klinotaxis circuit have been found to drive the BBC model of forward locomotion. It's only then that we start to test it under longer conditions, which is the graph that's below. So first, in conical gradients, instead of just running it for something like 20 seconds, which is what we did during evolution, now we run it for much longer, and we see that those initial bends in the right direction actually lead to the peak of the gradient. So we test it for generalization. And we also test it in different gradients.
One other comment to Netta's point. The reason it has to be re-evolved is that the feedback through the body and environment is now different, right? We've got a more complicated model here, and this just reiterates how important it is to think of the whole brain-body environment system as the unit of analysis.
Absolutely. So I wanted to follow up with two remarks, if I may, on specifically this point. So it's two of them. One of them is that as far as I'm concerned, until this paper, it was basically considered an insurmountable challenge to run extensive evolutionary optimization or any other sort of optimization in a variety of different conditions for a model that is neuromechanical, integrated neuromechanical with relatively detailed biology. Now, of course, this is an abstract model. The neuromechanical model is in itself quite abstract, and the steering model is abstract, and we're not denying that. We're quite happy with that, in fact, and we embrace that simplicity and that elegance. But even with the simplicity, it's actually quite computationally heavy, and so the ability to take that to the next step and evolve it, or in any other way optimize parameters, is a wonderful thing, and it's taking us a step forward, so I applaud that.
Can I make one comment about how that was achieved, though? I basically had to re-implement your model from scratch using sort of purpose-written code, purpose-written linear algebra routines, purpose-written differential algebraic integrator, and so on. We couldn't do it with the sundials-based one for a variety of technical reasons. So, and this, I think, speaks to some of the OpenWorm goals that we might want to talk about later. It wouldn't have been possible with a general-purpose simulator. It's just there's a couple of order of magnitudes that costs you that would have made this project completely infeasible.
You're absolutely right, and we had never actually intended that initial implementation to be a long-term implementation. This is a prototype which we're delighted that people have taken on and improved and re-implemented.
It's not even Boyle's code so much as it is sundials. Has a tremendous amount of, well, again, it's technical issues, but a lot of our ultimate problem had to do with sundials.
And the parallelism.
One of the things that would be a wonderful follow-up separately is to discuss that offline. I'm very keen to find out more.
The second point.
So the second point was, let me try and remember. So once you have re-evolved, of course the embodiment matters, and of course now that you've got a different model, you do. I completely understand why the model needs to be re-optimized. But an interesting question is, how do the two different models differ? And how do the parameters differ? And that I think we should spend a little bit of time on that. And if you could expand on that, that would be great. As part of that, maybe you could touch on the to what extent you trust the fitness function to just give you a great working model, and to what extent you think that's predictive.
About 2 points. So we're comparing the parameters and then trusting the klinotaxis index.
That's a good question.
In terms of comparing the parameters, the truth is that I've not gone in as much depth into this last one than we did the very first one, the simpler one. The thing
definitely on the to-do.
It's definitely on the thing that we have compared, and or the thing that I wanted to, and that I did report on that paper that I did think was important, was how the sensory motor transformation was or not similar. So more so the functional aspect of the klinotaxis circuit. If you give it an increase in concentration, how does that result in changes to the neck motor neurons in functionally, whether does it allow it to turn more or less? So functionally, the two circuits behave very similarly. But again, even from the previous work, I know that could be implemented in many, many different ways. And that was part of also actually, that was part of the result of the very first one, even without looking at the parameters of this integrated neuromechanical model. We do know from just looking at the different ways of generating the right functional sort of information flow in some ways. There are many different weights and parameter intrinsic parameters of the neurons, all of which can produce the exact same correct functional flow of information. So I guess that was part of the reason why I haven't gone into the parameters as much of that last one. But
So
yeah.
that suggests that indeed the vast parameter space and the vast multiplicity of different niches in parameter space that can generate the behavior should suggest to us that we might be learning something about the behavior, but not so much about the parameters.
At this point,
that is under constraint.
As experimental constraints improve, I would expect that to improve. And the other thing I'll make, the other comment I want to make is, if there's one take-home message from the information flow paper, it's that maybe, given this vast degeneracy at the neural parameter level, it may be that we need to be thinking about generality at other levels of description. And that's why I think it's interesting that we found a very similar, what we called information flow architecture across the full set of successful klinotaxis circuits that we evolved under the 2013 body model. Again, it's not the model per se that I would stake any reputation on, but I think that result is very intriguing, and it suggests that we might have to look at higher levels of abstraction to find sort of common principles, and that the level of electrophysiological parameters may just be too low a level, especially given the individual variability we talked about earlier.
Absolutely.
Cool. This is great. I'm loving this discussion, by the way, guys. So I'll chime in a little bit more here when I get to the end as well with some of my thoughts. But so thanks for the catch on the other things that needed to be re-involved. These figures were in the main line of the paper as well. That in addition to, I guess, re-evolving the circuit, was also just looking at which selection of muscles innervated by the minimal model were optimizing the chemotaxis index and the fitness, both in quantity and in identity. So those were more key parameters, I think, that went into exactly how to attach one to the other. Because, hey, if you have an evolutionary algorithm to maximize your fitness, why not why choose this a priori? Why not see what actually works well? So I thought that was pretty cool.
Anything that's not experimentally constrained, we have to study how the solutions vary when you vary that unconstrained thing.
That's right. That's right. I think that's pretty cool. Yeah. Yeah.
And so the main thing that we were trying to explore with this one, particularly with the bottom graph, was how important was it for steering to be able to modulate the dorsal ventral rhythmic pattern in the anterior most muscles.
Yeah, at the very tip.
So at the very tip. So, and what we found in this model, in this case, that you do need to modulate the most anterior segments of the body, not just halfway through the neck. That wasn't enough.
So do you have an interpretation for that?
Do we have an interpretation for that, for C. elegans, for the actual? No, not necessarily.
No, I mean, it's apparently a property of the way the body model works that this needs to be done.
Yeah.
So it pushes the question back to, is that feature of the body model accurate enough to make that a prediction rather than just a result about the model itself?
So I can expand on that a little bit.
Okay.
So in our body model, to make it hard for ourselves, we insisted that the model should work if the worm starts in a straight line configuration.
Mm-hmm.
And that's absolutely essential. Its previous models needed a sinusoidal configuration in order to make things work, and it was very important for us that the system should be able to start. And we also ran some very preliminary, very simple experiments on the worm themselves, on the animals themselves, to try and see what happened if we really straightened them up and then let them go, how they started moving. And the capillary forces holding the worms down and the passive cuticle forces and so on seem to be restricting the animal to not move as easily from the center of the animal. But of course, the ends of the animal are free, the tail and the head are free. And so it's only natural that when the animal wants to move, it's the head and tail that start moving. and then it seems to us that the animal is following the tip of the head or the tip of the tail, whether it's moving forwards or backwards. That's not to say that the head and tail can't move partly independently, but as the animal is progressing in some direction, it's going to be the head that's bending first and then moving everything else. And so when you're innervating something which is behind the head, without the head, you're imposing a mechanical load, which is just too hard for the worm to overcome. And so that would be my interpretation. The bit where I would stop short of is to say that the body is following the head. So the body is following the orientation of the head, but this would not be possible with a passive body. So the head is not strong enough to carry the weight of the animal, to drag it along. The only way this is possible is because the signal, the neuromechanical signal, is actually propagating, and the rest of the muscles along the body are participating and playing along with what the head wants to do.
Sure.
Yeah, that's a good specification. Yeah,
Very different. That's, I think, the key difference between the 2013 and 2015 implementation of your model.
Yeah, I think I used a little bit looser language earlier, so I think that's a really good clarification. Okay, so that was this part, and now okay, so these last two things are results as well that came out of here, and the key thing to understand this one is to understand, again, as we originally defined klinotaxis, it's not just movement towards a chemical gradient, but movement that involves the swaying of a head side to side. And so the tests that were done here later in the paper were showing essentially experimenting with stepping up the concentration of the chemical gradient depending on what phase of crawling the worm was in. So this sort of gets to a little bit of a, I don't think you guys explained it this way, but I sort of think of it as like a little bit of a mechanism. So I'm going to attempt to kind of show it. So it's sort of quantified here in this graph at the beginning, which is saying that on the x-axis, this is the phase of locomotion in which we're talking, and then they presented concentration steps either in the positive or in the negative, in blue or yellow. So there's the blue and the yellow lines here, and they're essentially showing, depending on the phase, how that's actually causing the worm to turn in terms of numbers of degrees. So if you're at the 0 of the locomotion phase and you get a positive concentration step, that's causing a pretty large turning bias. Whereas if you're down here, I guess, I don't know, what, 3 pi over 4 or something, just before pi, you're getting basically no contribution, and it's a negative contribution if you're all the way in the middle of the phase, and then sort of a similar thing in the negative on the other side. So schematically, what that means is that if I'm here at phase 0 or I'm here at phase pi, the black line here refers to the path that the worm would take if the ultimate sort of trajectory of the worm if there was no concentration step. And then again, the blue and the gold are showing the sort of equivalent path if you do get the step in the positive or if you get the step in the negative. And so what it's sort of showing is that if there's a positive concentration boost in the blue here, it's basically saying that in this configuration the worm is going to move down, and in this configuration the worm is going to move up. And that is kind of what you want because of the different differential positions of the head. So this is sort of summarized, you know, in this graph, which is basically showing how getting the stimulations that when the head is always up means that it's going to turn up towards the up direction, and when the head is down it means that it's always going to turn in the down direction. And so this kind of is a nice test to show that we're in fact getting klinotaxis that is actually differentially based on the position of the head. So how did I do? Did I kind of capture what this is trying to show, guys?
Yes.
Yeah, the only thing to emphasize maybe is that the C is based actually on experimental paradigm rather than...
Right, yes. So and the main point would be that C has been done for, I think, odor in the worm. So they started doing experiments where you can express a light sensitive protein in one of the chemosensory neurons.
Oh, right. This is like an optogenetics.
Neurons, I think other sensitive neuron. And then the main difference between C and B is that you're not perturbing the sensory neuron once, but you're doing it repeatedly at the same point in the phase, and that's why you end up observing not just a turn, but a ongoing turn.
That's right.
And so, yes, and so that I guess the main point to say is that there's a similar. I think there's a similar sensory motor transformation that is occurring in the model than it is in the worm. And it's equivalent. In this case, in the experiments that were done in the worm, I think it was, there may be things to do with, for example, sometimes the neurons, the sensory neurons are sensitive to attractant or repellent. So depending on that, the curves will be the opposite direction. Other than that, that's a consistent sensory motor transformation.
Cool. Yeah, and I think that's so that's an interesting thing that you can get, from combining, the models together. Of having these sort of different head sweeps that are present. Obviously, your other model did do, the up and down motion, but I think this is an interesting thing that you can see in this combination.
I mean, one of the points I wanted to make with this one, was that the sensory motor transformation was actually different, but qualitatively similar to the actual point model. And that was one of the points that I said earlier.
Yes, that's right.
That graph at the top happens to be, again, qualitatively similar. And such a graph has not been seen for the worm, or such an experiment throughout different phases has not been done for the worm, but that would be for any of the sensory modalities, chemo or other, or thermal. But it could be done. It has been only shown like in the C case, the CT case.
Got it. Great. Okay, and then the last one focuses a little bit, and this sort of gets into a space of kind of movement analysis that you're able to do here, where you basically did a bit of a shape analysis. And so this is going to be a little technical for folks that aren't familiar with the work of Greg Stevens in his eigenworms. But as a very high-level summary, essentially you can do mathematical analysis of the shape of the worm and determine you can sort of mathematically attempt to break it down into some more simple sort of shape configurations that seem to explain a lot of the potential shapes of the worm. And so what you're showing here in this last figure is a few things. First, you're showing this covariance matrix of the differences in the angles along the body of the worm. So your axes here go from the head to the tail, and you're obviously graphing them against each other. You're looking at the differences in that covariance matrix of the angle. So essentially, you can think of like each different joint of the original Cohen model as having its own angle. And my understanding of this is that you're comparing the change in angle from one point to every other point in this covariance matrix. Is that, do I have that initial part about right?
Yes.
In terms of what this is showing? Okay. And then you're comparing two conditions across all these subsequent pictures of just forward locomotion, meaning that there's no steering involved, so there's essentially no contribution of your minimal klinotaxis network, and comparing that to the case where you are actually steering. And so you're looking across these two conditions. And so the covariance matrix, you can see there's some qualitative difference, just in terms of the width of this area. You go in a bit further to kind of dig into what that might mean. And so you're showing that actually, okay, so in terms of eigenworms, and again, forgive me for those of you who don't have eigenvalues at the top of your mind, but I think most folks in this audience do right now. So this is a graph showing essentially these principal components that come out of doing the this movement analysis. And what you're basically showing is that if all the model is doing is forward locomotion, you can basically explain that with a smaller set of eigenworms or principal components than you can if there's steering. And what you're basically saying from this result is that when we introduce steering, there's essentially more, how to say it precisely, there are more eigenworms that explain the variance that are involved, that you would kind of need in order to explain more of the variance. So the red here is, again, the red dots are showing the ones from forward locomotion, so it's showing that you're putting a dot here at explaining 50% of the variance, right? And then you pop up to 100% of the variance here, whereas with the blue, the first one only explains about 30% of the variance, and then in addition to that, you have a much smoother drop-off before you kind of explain, before you get up to like 80% of explaining the variance. Do I have that about right?
Yeah, that is correct. Yeah.
Okay. And so then you further sort of show that here in terms of overlaying the contribution. So this is a graphical view of what the eigenworms actually look like. Again, from head to tail, sort of angle posture. The first one looking like this, and they look pretty similar for both forward and steering cases. But then the ones that are more relevant for steering start to look a bit different. So these just kind of look like sinusoids, whereas this actually has a bit of an asymmetry to it, which is interesting for steering. And then you're overlaying those, you're sort of representing those eigenworms on top of each other and how they look different for forward and steering to each other. And then maybe if you guys want to sort of say the bigger point, the bigger takeaway from all this for you guys would be helpful.
Okay, yes. So I think the main point that I wanted to say here, this is a little bit more exploratory. I think the main point is that having an integrated neuromechanical body to study steering means that we can begin to compare it with how the worm moves and how the worm does steering a lot better. And that includes, there are different ways of characterizing the behavior of the worm, but one of them that has gained popularity is, for example, doing this kind of shape statistics. So the main point is that whereas before with the point model, we couldn't do any kind of comparison with how the worm steers. Here we can begin to do some of those comparisons. And so this throws one idea. So steering alone hasn't been studied with the shape statistics, or not a worm that's just steering. And so this begins to throw some ideas about what how the shape statistics would change. But it's not, I wouldn't say it's very conclusive yet. One of the it's very puzzling to me, and I haven't figured out entirely why some the eigenworms wouldn't. There were so many eigenworms that define the behavior. So that's something that I need to continue to explore. But one of the interesting things was that the third eigenworm, so the first two are obviously very related to just the actual forward locomotion as before, but the third one is most of the variance that is decreasing is in the head, which is related to steering, obviously.
Ah, right.
And then you can see that in the green part of D. So the third eigenworm in green is reducing variance mostly about in the head. But again, that is just one step into looking at a full body that is steering.
Right.
Maybe I can chip in here a little bit with a bit of a caveat.
Sure.
About neuromechanical modeling. So every model makes assumptions, and in our case, we modeled the VB and DB models as bistable, and consequently, the muscles were effectively on and off. And so, when you have effectively or close to binary systems, so we also implemented a sort of graded potential model which had some bistability in it, but it's close enough to being on and off. And so whenever you have something which is more or less binary, you don't have a lot of freedom in modulating amplitudes. And what you need to be modulating in order to affect steering is the duty cycle, or the timing of switching on and off. There are other implementations of steering models where what you're modulating is the amplitude. And there are other models where you might be modulating both. And so presumably the effect on the trajectories you see and the waveforms that you're going to see are going to be quite different. And I think that's an interesting avenue to go down and try and understand much more closely what is the actual sensory motor coupling, what is the feedback, and how does that translate to waveforms, and what are the advantages and disadvantages of different implementations.
Yeah, as Eduardo said, I think the key point of this little result was just to open up a new source of experimental constraint into these kind of models.
Yep.
Yeah.
That's right. Okay.
And I wasn't saying this as a criticism, but as a sort of looking forward to
Absolutely.
exciting stuff.
Absolutely. Awesome. Okay, so then so that concludes. So that was the final sort of result figure in the paper. So now is a sort of section for discussion. Although we've been having a really robust discussion already, it also kind of validates that I think I was right. I probably at this point could not go through all the figures in the information flow paper, although I lament it because it is actually really interesting, and I recommend if you're watching this now and you get a chance, you should really go through that one as well.
I end with one comment that we couldn't have done anything like this if we had had to try to build our own neuromechanical model of locomotion from scratch. It was really important that we were able to build on what Netta's group did, and the fact that they made the code publicly available was a really important resource when we rebuilt the implementation for Evolution. So I just want to acknowledge that really important contribution.
Thanks. No, we're in general very supportive of everybody who makes code available, and we think that C. elegans is a challenging enough problem that we can all work on it together and help each other. So
yeah. Well, ours will be available too as once the final paper journal version of this work gets submitted, and we'll also be making ours available.
Fantastic.
Well, and I want to congratulate both you guys, both teams, which I know it's a group effort of many people. From our perspective, and now speaking a little bit wearing the OpenWorm hat, I mean, we came into this, I think later than most of you, in 2011. We were sort of looking around, and we found Netta's model pretty quickly from Jordan Boyle's, dissertation and that previous work. I'm getting more and more familiar with some of the other background that leads into this. But what's exciting as well about this paper is how many of the things that have happened only within the last 4 years are now integrated in here. I mean, eigenworms, that was 2013 as a big one in terms of incorporating that. Some of the data that's been coming out from the optogenetics that you're able to do here, some of those experiments really were just starting to come out in the last 4 years or so. And then, reusing this model within that span of time as well is very exciting. And, I think this end-to-end vision of having a neuronal network, filling in the gaps with, some sort of evolutionary computation, having a neuromechanical model, and then comparing it with, results from actual movement analysis, it sort of is the trajectory that, we from OpenWorm as well imagined from the beginning. And I think that what's also exciting is that you guys have been able to execute the full loop, relying on, of course, assumptions. But I think that's a really exciting point. I mean, I think when we began, we started being excited about all the levels of complexity, beyond which I think you probably wisely initially have, put aside. So things like ion channel dynamics, which I think are important at some point. Or even more intense, physical simulations of the body of the worm, which we've been working on, and this community has been working on as well. But I think what just some applause for this particular paper. What's exciting is that those practicalities of making the model work very rapidly, and having a model that's minimal enough on the neuronal side that you can put in that same loop and apply all that to evolution, I think is a really exciting existence proof. And I just wanted to, just to echo as well on top of Netta, how, I think you're right. Before this paper, I don't know that folks would think that you could go that entire loop. So I think it's extremely exciting and stimulating, for the field going forward. So some discussion points that I had that I wanted to take things to the next level for all of us. Some of them are sort of sync and coordination points, but there are a couple scientific ideas that we started to touch on, but I think kind of go to the heart of what the next phases are. And I'm really curious what your guys' impressions are. I think one that this idea of fully determining behavior via circuit versus global network dynamics. So we got pretty excited, and we've become increasingly excited, and we've had previous journal clubs on these experiments that are looking at dynamics across a lot of neurons. And Shrek was actually talking about some of the experiments that he's doing with this in his lab. But I think there's this really sort of foundational assumption about the boundaries of a circuit that's responsible for a behavior. And I think you've touched on it in a few different ways, talking about minimal versus maximal versus intermediate versus multiple models and all sorts of things. But I guess to sort of say, like in a point, like what happens if, in fact, like klinotaxis is actually, really does have contributions of, dozens of neurons, and that the information flow really is, happening across, dozens or a majority potentially of the neurons. This is actually a bit of a plug for we're going to have we're going to do this journal club paper that Saul Cato put out in Cell. We're going to do this as a journal club in a couple months. And have Saul join us, and as well as other folks from his team. But this idea of sort of global network dynamics, I'm just interested in your guys' perspective, both Eduardo, Randall, and Netta, about kind of what happens and how do we go from these minimal network models to trying to work with potentially almost even the whole connectome.
I think that's what's exciting about C. elegans, is that the gap is not nearly as large as it is in pretty much every other nervous system that people are looking at. You're talking about dozens of neurons to hundreds of neurons. That's a big gap, but it's nothing like billions or so on. So I think the only way in which the strategy that for example, we're trying to pursue would be completely discounted would be if the contribution of everything is equal. And I don't think that's the case. I mean, I think you can find, it's almost like an eigendecomposition. I think you're going to find that certain parts of the variance, certain parts of the behavioral features are going to be explainable by the simplified, reduced models, and there's going to be a residual that has to be dealt with, and that's going to push you to incorporating more of the circuitry, more of the interactions, more of the complexity. And ultimately, I would hope that you'd be able to say something interesting about the entire nervous system. That's an ambitious, somewhat ways off goal, but I think in the C. elegans community, it's much more feasible than it is in any other animal system that people are looking at this in.
And yeah, if I can add to that, so the way I see it too is, so part of what we're very much wanting to do is to integrate neural circuits that do slightly different behavior. So particularly, for example, in the case of taxis or spatial navigation in the worm, there are so many different modalities and strategies that it uses, all with overlapping sets of neurons and other new neurons. And there's more than one just set of neck muscles. There's a number of different neck muscles and so on. And all of them will be very much working together and or in complementary ways, or sometimes, anyway, all of the different ways in which all of those networks will need to work for the worm to orient spatially will begin to give, as more and more of these behaviors get integrated, I think you can build a bigger and bigger picture of all of these different neurons doing things. And in the case, the integration that we did now with Cohen's model, Netta's model, was less of that, because the sensory neurons, the neurons in the klinotaxis circuit weren't interacting in any meaningful way with the with the rest of the motor neurons. But hopefully, the next set of integrations will be dealing with more of that. I think that's the goal.
Yeah, so I completely agree. Sorry, did you want to say something?
No, go ahead, Dr. Cohen. Go ahead.
So I can completely agree with that. I think that I can add to that, first of all, that it's an incredibly exciting time for all of us to be witnessing the remarkable technological progress just of the last few years. Every few months, there's massive progress in being able to look for longer time and under more scrutiny and different behavioral assays, looking at more neurons in higher temporal resolution, higher spatial resolution. We're beginning to see possibly even multi-compartmental dynamics. It's all really, really exciting in terms of then hoping to try and get a big picture of this one considerably simpler system than indeed the others. But from the modeling perspective, what we're trying to do, all of us, I think, is to build simple models of really complex systems. And yet we're trying to make these simple models in some way realistic, in some way predictive. And we need some humility there. We all, I think modelers have that humility, but sometimes it doesn't come across and to the community at large. What people modeling in complex systems very often face is that they have some system and they have some data and they have some behaviors and they put them all together and they build a model and the model works great. And then you add the next bit of data and you test it against it, or you add yet another behavior or some other measurements, and everything falls to bits. And so the ability to take a complex system and add to it in an intelligent way, in such a way that it doesn't fall through and doesn't all just shatter in your face, is the challenge that the complex systems community is facing. And that's not an easy challenge. And I think what you've done here in being able to put together two aspects of this is already a step forward. I'm not arguing that either one of those models is actually the right model, but the purpose of a model is not to be right. The purpose of a model is to be falsified. And the only way we can falsify a model is if we're incredibly strict and precise about our assumptions. And so I'd like to say one more thing, which is I think in this paper of yours, you were incredibly clear about what the assumptions were. And that's really important for the discourse and for that iteration of experiments and theory. Be really clear, these are my assumptions. Here are the things I can falsify. Here are the predictions given these assumptions. Here's what the model is purporting to say, and here's what it's not purporting to say. And then exactly what you said, Eduardo, if we look at sufficiently many behaviors and say, okay, can we build a framework that's going to capture as many of those behaviors as possible, as many of those modalities as possible, maybe that'll give us a bit more confidence that the model can say something about the real system.
Great.
Excellent. So I think I think that's really interesting, and I think you know, there were a few different pieces that were in there in terms of Netta talking about you know, simple aspects of complex models. This idea of you know, which neurons are kind of driving a behavior and which ones maybe are more on a residual. You know, from Randall as well and Eduardo, and you know, I didn't really explain the picture that I put up. But you know, it's coming from looking at the worm not doing anything as an activity graph of time, you know, from left to right, and showing sort of a lot of neurons being active even when the worm's not doing anything at all. For me, and you know, we're going to dive into this more when we do the journal club, but for me, you know, it's almost potentially doing a fundamental reversal of figure and ground. Where the concept of a behavior as this sort of feed-forward activity, which of course we know again is an assumption, you know, all these are assumptions, but the idea that like a behavior sort of comes in and it information flows from one step to the other, and then there's a result. This idea that there may be dynamics happening across all these neurons all the time, even when the worm's not doing very much, means that there's like a baseline, like a carrier frequency kind of a dynamic that's happening in the worm at all times. And then as additional information flows in, it is modulated by the other activity that may be going on, and it's some sort of a combination. So I think that it's consistent with the kind of mental model of what you're saying here, where there's like a behavior and then there's a residual. The question is, when you go back to the experimentalist and we say, here's a prediction that we have, about these things, how much will we need to incorporate the activity of the other neurons that are outside of our minimal network? Even if you make the minimal network bigger, right? How do we do that? How do we do that appropriately? And also, how do we do that in a way where we don't necessarily blow up our computational demands? I think these are all sort of good questions. I don't know that necessarily we have answers for that, but this is just sort of my thinking as we move from this era of this sort of just pure circuit type ablation experiments to these like we can look at all neurons type experiments and look through patterns.
I certainly encourage the picture that you're describing of the nervous system as a dynamical system, which is effectively what you're saying there. And again, the whole point of the sort of brain-body environment system perspective is that the nervous system itself has dynamics, there's feedback through the body dynamics back onto that neural dynamics, and there's feedback through both of those through the environment back onto that dynamics. And I agree with you that seriously confronting that picture really changes your sort of feed-forward view of how behavior works. And I will comment that one of the assumptions that we talk about in the information flow paper is that we explicitly cut that feedback loop through the body and environment and just looked at how imposed sensory information modulated the motor output. And it's much more difficult to see how you would push through analyses like that when you take seriously the sort of coupled dynamical picture of the whole system. But I think that's the right one.
And it is something that we're trying to look at and trying to do. Yeah. And also, I wanted to add to the Cato paper, which is really exciting. We're very excited about it. The worm is very much trying to, obviously, it's locomote forward and locomote backwards. It's not being able to. And that's a little bit what they're trying to see there. But it is trying to locomote forward, trying to reverse every once in a while, trying to go backward, trying to do omega terms. So that's why they, and you guys will discuss it more, I suppose. So that's why you see that kind of idea.
Yeah, that's a good point. Yeah, it's not just
dreaming.
Doing nothing.
Yeah. Okay, great. So then, we don't have a lot of time, and I do want to stick to to the time that I promised you guys. But I did just want to make a couple comments about the sharing. So as we saw in the paper, this involved the combinations of code for multiple projects, and you guys even say in the end of this paper, I think a lovely thing in your future directions is sort of saying that we'll need to do more combining of different models together as we go forward. And so this is very much, the vision that we've had in the OpenWorm community. And, Netta's been familiar with us from the start because we reached out to her very early, and we've been very grateful for the contributions that she's made. And we hope that we want to be a good actor in this community. And as there are additional, groups that are building on this, Eduardo, I know that you're building out your lab now, and there's new folks that are kind of coming into the picture. Just wanted to say some of the unique things that OpenWorm can help to bring to the table. So we largely have outreach in in addition to the scientific community that we're talking to, and I go to meetings and participate on the science and that kind of thing, and we definitely network on the science side. The other side of this is to bring in talented programmers who otherwise are not necessarily familiar with the C. elegans or the worm, but they're excited about the idea of solving this challenge of how do neurons give rise to behavior, and how does that relate to brains, and to think about that in terms of consciousness. So folks come in curious about how to do open source contributions, and they come in with some best practices from the open source programming world, things like using open code repositories and tests and these sorts of things. And so the space that we've sort of tried to occupy is a space where we're this nexus point between these scientific questions and the kind of explorations and experiments that you're doing, shared code bases that are made available for everybody to see, which is also good for reproducibility, and then the sort of talent that's out there that otherwise might contribute to working on the Linux kernel, but instead comes in and maybe helps to flesh out a database of data that we can all reuse and build sort of shared experiments around. And I think that it's a really good time, as we're starting to see some consolidation of some of the best-in-breed models that have been around for a little while, to just sort of to talk about how those things can be shared more openly. I would also say, in addition to OpenWorm, that we also have other partners that care about sharing in general. So the Open Source Brain, for example, is a resource out of the University College London that's generally for sharing neurocomputational models. It doesn't get so much specific into the worm stuff, but there's an overlap there. So this idea of sort of sharing is one that I think can benefit everybody and benefit you guys. And also, I guess the last thing I'll say is that we have folks at the software engineering level who think about how you'd scale a model in general that is going to continue to have more and more and more pieces and parts to it, which goes beyond sort of everybody writing C++ scripts, which are excellent, at the beginning. But as we're going to mature, I think, going forward, I think we're all going to see the demands on the code getting more intense. I think just naturally we're hitting the limits maybe. I don't know, you guys tell me your experience, but we may start hitting the limits of what we can do with just everybody kind of writing their own scripts. So while we probably can't go into the exact details of what next actions we might take here, I just thought I'd put that out there and let you guys know and say that there's the potential for, I think, even more effective collaboration with code, around and around methods, and we'd love to help be a part of that as OpenWorm.
One thing I wanted to mention, Randy, you may want to mention it, but so the evolutionary algorithm and the neural network, so the neuron model that we've used
It's modified that we haven't released that. Yeah, so it's
okay.
mostly the search right
At least
now.
the evolutionary algorithm, the search procedure is on Randy's website, so you can find that online. And some of the other types of code that we need to do need to be online soon.
This is the stochastic optimization.
The
Yeah.
stochastic optimization is, yeah.
Our version of your body model and the neural code will be released, like I said, once the paper is.
Fine, yeah.
Yeah.
The neural code, in some ways, is very similar to what it
what's
is,
there.
except for the gap junctions.
Except, and the sparse connection matrix.
Yep.
Yeah.
Right.
But yeah, we agree very much.
So, because the next enterprising scientist that wants to build now on your guys' work, this combined thing will want to be able to have a good way to make sure that they're not missing anything that you've added in. So how from we don't want to be in any way dogmatic. If we can help when you get to the point of sharing some of those things, we can we set up other labs on things like GitHub. For example, in order to follow some best practices, so we can help with that. And also, we some of our resources, we've been aggregating the latest. So for both of you guys, we've been aggregating the latest information from Scott Emmons and Stephen Cook on the connectomes, and that's something obviously is probably easy for folks to get a hold of. But we've been building a layer of Python code that makes it so that you can just write a little bit of code to pull out the connectome rather than everybody reparsing the CSV files. And so things like that, we want to be helpful. So as much feedback as you can give us as well, if there's room to help or things that we can do to make your lives easier in sharing code with each other, please let us know.
Yeah, if I can just throw in one very quick observation, GitHub is definitely a great place to be putting your code, because not only is it making it publicly available, but also if you make any changes to the code subsequent to the first initial publication of the code to the public there, you can track those changes and people can see on an ongoing basis what the differences are.
Great, yeah. I want to add one more thing too, Stephen. The other tool that we've been sharing and making available is that, so the connectome searcher that you talked about at the very beginning of your presentation, where we were trying to explore which sensory neurons connect to which neck motor neurons and so on. there is a tool now online that I mentioned to you when we met in Chicago, which is in the link, in the URL, elegance.heroku app. I can send the link, or we can put the link up.
Right.
That's a tool for searching for subsets of neurons within the connectome, which again, I think is still a tool that is the only tool that exists of that kind.
Awesome.
You guys are very welcome to incorporate us in the way of links or in any other way from the OpenWorm project.
So maybe I can add two more things. One is specifically with respect to the connectome. There's been the reanalysis of the connectome and the worm wiring webpage, and so it's important to keep in touch with which data set you're working from and whether that's important or not important for the specific work you're doing. Specifically for shortest and strongest path, I expect that the original White et al. And Varshney et al. Revision of that would have yielded very similar results to worm wiring, but in general that may not be the case. And so I'm not sure whether the website that Eduardo just referred to feeds on the updated data.
We use the Varshney 2011 data. Is that
that's not the latest? Yeah. So. So that's one thing, but the other thing that I wanted to say is, Stephen, you were asking how you can help and so on. So I wanted to emphasize that even just these open seminars are opening things up to the community, and so people can then contact and say, look, I'm interested in that. Can you show me this code and that code? And in my experience, people are always happy to answer questions, always happy to respond. And so thank you for making these four available and for all the service.
My pleasure. My pleasure. Thank you all for spending the time, as we couldn't do it without you guys. So, okay, great. Well, I think we can take other conversations offline. If we've had about consistently about 6 or 7 viewers have been watching, and obviously it'll also be up on YouTube after this. So. If you've made it this far, thank you so much. And you've now seen the folks responsible for the work here. And I would echo Netta to say, reach out to them directly. If you need a connection, you can always reach out through all the OpenWorm channels, and we can put you in touch. But okay, so I will draw it to a close here for time. Thank you so much for participating, everybody, and we'll see you next time.
Great. Bye-bye. Bye.
Great talk. Thanks, guys.
Bye.